Hello guys, we are the students of Shivajirao S. College of Pharmacy, Asangaon. We have done a project on Agremone mexicana Linn herbal antifungal cream, which is an antifungal cream which acts on fungi to stop their growth or kill the fungi. So before the formulation of the cream, we need to know what is a fungi. So fungi, the word fungus is derived from the Latin word for mushroom. Fungi are unicellular or multicellular eukaryotic organisms that exist in all environments worldwide, from fungi visible to naked eye, such as mushroom, to microscopic yeast and molds. Any diseases caused by fungus called mycosis. Fungal infections are mostly like to affect the skin, nails, or lungs. Fungal diseases kill more than 1.5 million people worldwide. As you can see in the picture, these are the multicellular structure of fungi, and here is the unicellular structure of fungi, which is also called as yeast. So after the structure of the fungi, we are going to study the species causes fungal disease. Fungal infection is a man divided into two main groups. First is superficial infection, second is systemic infection. First, superficial infection, example microsporum, trichophyton, and epidermophyton. And second one is systemic infection, which is asparagus niger, candida albicans, cryptococcus, blastomyces species. We are going to formulate the cream on the dermatophytosis, which is also known as a ringworm or tinea, caused by a group of organisms capable of invading keratinized tissue such as stratum corneum, nail, or hair. We need to understand what is a topical drug delivery. A topical drug delivery system is a way to deliver medication that is applied onto a particular part of the body, typically the skin, to treat various ailments. There are many common forms of topical medication such as lotions, gel, patches, and powders, but they are mainly formulated as a cream or ointments. Advantages of topical drug delivery system are alternative forms of oral administration, reduce hospital congestion, they can avoid gastrointestinal drug absorption difficulties. Disadvantages are skin irritation, contact dermatitis, as well as allergic reaction. Those drugs having large particular size, they do not easily absorb in the skin. Now we will study the physiology of skin. The body's largest organ, skin protects against germs, regulates body temperature, and enables touch, tactile sensation. The skin is multi-layer organ composed of many histological layers. It is generally described in the three major tissue layers. First is epidermis. Your epidermis is the top layer of the skin that you can see and touch. Keratin, a protein inside a skin cells, makes up the skin cells and along with other proteins sticks together to form this layer. Dermis. The dermis is connected to the epidermis at the level of the basement membrane and consists of two layers of connective tissues, the papillary and the reticular layer, which merge together without clear demarcation, composed of strong connective tissue containing collagen and elastic fibers. Hence, it can easily stretch and recoil easily. Now we are going to study the extraction, formulation, and evolution of Agremone mexicana Linn leaves as a herbal antifungal cream, which is our main project. So objectives are to carry out pre-formulation study, to provide an overview of fungal infection, to formulate antifungal cream. As you can see in the picture, these are the Agremone mexicana Linn leaves and plant. Introduction of Agremone mexicana. Medicinal plants are believed to be an important source of new chemical substances with potential therapeutic effect. Therefore, this plant should be explored for the discovery and development of newer and safer drugs. Agremone mexicana peperviaceae is commonly known as Mexicana prickly poppy in English. In Marathi, it is called as Ferangi Dhotra, Bilayat, and in Hindi, it is called as Satyanashi. The plant is self-sterile and predominantly self-pollinated that prefer light soils, requires well-drained soil and can grow in nutritionally poor soil. It cannot grow in the shade. Various parts of the plants have various medical effects and uses. Argemone mexicana is reported to have different chemical properties such as anti-hepatotoxic activity like antibacterial, antifungal, anti-HIV and wound healing. The plant contains alkaloids as berberine, protoprine, sanguine, optimized glucosides, terpenoids, sternoids, and tannins. Mexicana leaves and stem contains the strong antifungal property against the flammatosis fungi, namely Mucorindicus, Aspergillus flavus, A. niger, and P. notatum, Candida albicans. A Mexicana stems and leaves are equally soluble in water, methanol, and ethanol. Argemone mexicana is an erect spiny annual and biennial herb up to 1 meter tall with a slightly branched taproot. Its stems is branched and very prickly. It exudes a yellow juice when cut. These leaves are thiced like and alternate, without leaf stalks, toothed and with active pharmaceutical ingredient of our plant is S-chlorine. S-chlorine is berberine alkaloid isolated from Argemone mexicana. It is a plant metabolite. It is an organic heterocyclic compound and berberine alkaloid. Source of S-chlorine is opium poppy, Crodon flavans, and certain plants in genus Erythrina and some genus of Peperoaceae. Mode of action of S-chlorine is berberine is a co-product of S-chlorine. Treatment of fungal cells resulted in dysfunction mitochondria, which was evident from its slow growth and non-fermentative carbon source and poor labeling with mitochondrial membrane. Potential sensitive probe. This shows the fungicidal activity of S-chlorine. After the mechanism action of S-chlorine, as our plant shows antifungal activity, so we need to perform some test on it. So for that, we need to formulate a herbal extract. The herbal extract is prepared by cold maceration process. Leaves of Argemone mexicana are collected and washed with distilled water. After that, the leaves are shed dried for 14 to 15 days and then powder the dry material separately. 10 mg of powdered leaves are soaked in 30 ml of 99.8 by volume ethanol and kept for maceration for about 3 to 4 days with occasional shaking. After a couple of days, filter the content of conical flask by using filter paper in the beaker and transfer the content into china dish. Take the china dish over the boiling water bath to evaporate the sample and dry the ethanolic content completely. After drying, remove the china dish from the water bath. You can see the solvent is completely evaporated and the ethanolic extract is completely dried. Then add sufficiently quantity of dilute HCl to dissolve this dried residue. After dissolving the dried residue, filter all the content from the china dish by using the filter paper and collect the liquid extract below into the beaker. So now the extract is ready for the alkaloid test and for the preparation of the cream. As our plant is showing alkaloid presence, so we are going to perform some alkaloid test on it. First, we did a drag and drop test. By adding 1 ml of drag and drop reagent to 2 ml of extract, an orange-red precipitate was formed, indicating the presence of alkaloids. Hager's test. 1 ml of extract were treated with a few drops of Hager's reagents. A yellow precipitate was formed, indicating the presence of alkaloid. Wagner's test. Add few drops of Wagner's reagents in test tube containing 1 ml of extract. The reddish-brown precipitate is. After performing test for alkaloids, we have performed antifungal test. For that, we take a slice of a bread, sprinkle some water on both sides of bread, place a bread slice in a closed container, wait for 3 to 5 days. After 5 days, we can observe clusters of tiny plants on a bread slice. Mold asparagus is a type of fungi that grows on a bread. After growing the fungus on a bread, add extract on the bread. After adding our extract of Agremone mexicana, wait for 2 days. The results comes after 2 days shows our extract is able to kill fungus and having antifungal activity. Results of the antifungal test showing the API is able to kill the fungi and the bacteria in the cultural media. After the antifungal test, we are going to formulate a cream formulation. So for that, this is the ingredient table we have needed to formulate an antifungal cream. So first we need to prepare a oily face. For that, beeswax, white soft paraffin, liquid paraffin, hard paraffin and borax are accurately weighed and melted in a porcelain dish. By keeping in a hot water, the temperature is maintained between 65 degrees Celsius to 70 degrees Celsius of oily face. Preparation of aqueous phase. Water is heated and the temperature of the phase was maintained at 65 to 70 degrees Celsius. Development of cream formulation. Aqueous phase is slowly added the oily phase at 65 to 70 degrees Celsius and mixed for 10 to 15 minutes. The herbal extract is added into the above part slowly when temperature reached to 40 degrees Celsius, mixed until it get cold. The pH of the temperature is kept between 5 to 7 to matches with the skin pH. After that, we have performed pre-formulation studies. Pre-formulation testing is a first step in the rational development of dosage form of drug substances. Pre-formulation testing can identify or defined as an investigation of physical and chemical properties of drug substances alone and when combined with excipients. Objectives of pre-formulation study are: the overall objectives of pre-formulation testing is to generate information useful to the formulator in developing stable and bioavailable dosage which can be mass produced. The formal formulation study should start at a point after biological screening when the decision is made for further development of the compound in a clinical use. After pre-formulation study, we did evaluation test of mexicana antifungal cold cream. First, we did homogeneity. Homogeneity is the formulated cold cream was tested for homogeneity, visual appearance, and by touch. After the feel, emulsions, slipperiness, and amount of residue left after the application of fixed amount of cream was checked. Type of smear. After the application of the cream on the skin, the type of film smear formed on the skin were checked. Removal. The base of a removal of a cream applied was examined by washing the applied part with the tap water. Then we did a sensitivity test. The cream which was prepared has applied on 1 centimeter of skin hand and exposed to sunlight for 4 to 5 minutes. The above picture shows the before and after results of the sensitivity test of Mexicana cream. Then we did a pH. The pH of antifungal cream was determined using pH meter, the most accurate common method for measuring pH using the lab device of a probe and meter, or simply pH meter. Measurement is made by submerging the probe in a semi-solid until the reading is registered by meter. Results and discussion. Physical observation. Parameters are color, odor, test, states. We did 2 formulations. For that, color is white, odor is pleasant, texture is smooth, and state is semi-solid. Washability observation. Both the formulations are easily washable. Sensitivity observation test. For both formulation, no irritant effect, no erythema, and no edema observed. pH of the formulations. For formulation 1 is 6 and formulation 2 is 8.2. And all after that, we have concluded that our plant Agremone mexicana, which is from family Papyracea, it has antifungal activity against many species. Ethanolic concentration of Agremone mexicana extract were prepared to measure the activity against fungi. The cream then evaluated under different conditions. The evaluation tests and physical tests includes pH, viscosity, homogeneity, odor spreadability, and the antifungal assay. And therefore, we have concluded that our formulation 1 is more effective than formulation 2 as the pH of the formulation 1, which is 6, is matches to the body pH. Thank you.